All right. Okay. I think I'll just talk until Sam got his thing up. Uh, okay. I hope you guys are not tired of this RNA things. But this in the last couple of months, I was working on them, and I still don't know what I'm doing. Okay. So whatever I say, take it the salt of grain and with a grain of salt. Okay. Okay. The summary. I talked a little bit about microRNA, which I've talked for the last couple months, and but I will show you what the species or organism that I have studied. Okay, one thing which at one time I was very excited that I found lots of microRNAs in mitochondria. Okay, so but then it kind of doesn't seem to be anything. But I think there's still something in it. And then I try to open up the huge genome files and try to look at them. So these genome files are basically more than 100 megabytes for each. And I was able to open them up and look at the things and search out things that I want to look at. So it's a pretty useful thing if you are in the field. Okay, then I will show you some findings that I have on the human chromosome 1. Chromosome 1 is the biggest chromosome in the human genome. So if I can handle that one, I can handle anything, basically. And there's also lots of interesting things in bacteria. And I was looking microRNAs in bacterial genomes, and I couldn't find any. They're not there. So, but I'll tell you what I found. And the last, I will just talk about the tools that I've been using. So this is just an introduction.
Here we are.
Okay. All right. Yeah. Okay. So, but I believe the microRNA has much more important functions in the molecule, in cells. So what I have, that's for 20 years, people have studied lots of microRNAs, and there's an organization which collects all the data and then publish them on the web. So miRBase is the database. The latest one, or 3 months ago, I downloaded, and it has 48,000 microRNAs listed in it. And so I look at it and found that there are lots of duplications because many microRNA are collected from different species. And many species share the same microRNA. So if you delete all the duplicates, you have 27,000 unique microRNAs. And in that collection, 5,000 of them are from human. Okay? So this is the base that I want to do my analysis. So I think I have lots of microRNAs, and I can use this base to compare with other genomes to find out if genomes have how many of them each genome has, and how they are arranged in the genome. Okay, so these are many organisms that I have looked at, and I got all the data in my computer, so I can call them out anytime that I want. So the smallest one is mitochondria. Mitochondria exist in most of the species. They are inside a cell, but it has its own unique genome. And Nashua, this is the smallest living organism. It's a single cell, what they call the arc bacteria. That's the bacteria older than the current bacteria. And Ruda is another one, which is smaller, a little bit larger than Nashua. And Megan is the one which, if you remember, Craig Venter, who was working on who was the first guy decoded the human genome. Okay, and he worked on this particular organism because this is the first freestanding single cell organism, and it's the simplest one. So he actually synthesized this in the laboratory. So that was a very big deal. And E. coli, there's a bacteria, and nematode, that's a little flat worm, and it is very interesting. It's the simplest animal, and people have traced its development and found out that the adult nematode has 960 cells, and they can pinpoint each cell in the body, and they can trace each cell throughout the development stages. Okay. So this is the one organism that we have most data on, and we have the best understanding of. And then I download human, and with human I also download mouse, and just to see the human and mouse are very close, very closely related. Okay, believe it or not. So these are the organisms that I've looked at, and here is a summary of these their properties. So mitochondria in the human, it has 16,000. That's a complete genome, okay? And that's the smallest complete genome we have found. And there's 37 genes. And Nashua, it has 100,000 base pairs. And Ruda has 159,000. And then the bigger one is these are parasites. These two are parasites. They cannot live by themselves. They have to be another animal. But Megan is the smallest freestanding organism. In order to freestand, it needs 580 kilobytes or kilobases. And E. coli, which is a very well-known, well-studied bacteria, and it jumped to 4 million. And the nematode, which is an animal, which is a multicell, the simplest multicell animal, has 100 million base pairs. And the mouse has 2.7 gig. Human has a 3.289 gigabases. So these are the things I have downloaded and I have analyzed them. And so here, these organisms has only one chromosome, and these chromosomes are circular. And then the animals, the simplest animal has 6 chromosomes. Mouse has 21, and human being, we have 23. So what I'm doing in the last 2 months was try to analyze, to search through the genomes of all these species and see how many microRNAs I can find in there. Because in the last presentation, I asserted that microRNAs are like subroutine calls, subroutines in computer. So it's a high-level code, and level code will be should be a sequence of subroutine calls, or in force we call tokens. So I'm looking for clusters or lists of microRNAs, and I found many of them in the what they call non-coding RNA. But since non-coding RNA are derived from the genome RNA, well, I just go back to the genomes to find if I can locate these microRNAs and see how they distribute in the genome DNA. So this is my belief, okay? So the in mitochondria, I found several in the human in the human mitochondrion, I found several micro RNAs. So that I think since I found them in the mitochondrial DNA gene, they must serve certain functions. Okay, what is their function? I don't know, but by the presence of these microRNAs in mitochondria indicate that they have functions. And this is the in a human mitochondria, I found 5 microRNAs, and these are the sequences. At this point, I'm not expert, so I really don't know. I can identify all these microRNAs, but really it doesn't make any sense to me because these are just numbers. But for geneticist, he might know better than what these microRNAs, what their functions are. But my understanding, or from Google, not many people understand what microRNAs are doing, except they can inhibit the process of messenger RNA. So this is what I found, and once I found them in the human mitochondria, since I have a collection of all the mitochondria for many different species, they are also collected in a big library, and so I can go there and pull out all the mitochondria in different species, and then compare and try to see if the same microRNA in the human being exists in other species. So microRNA generally appears in mitochondria of all species. Okay, But from my analysis, human and mouse share most of the microRNAs. But then other different species have different microRNAs. And here is kind of a summary that I analyzed all the existence of all the microRNAs in all the mitochondria you find in these different species as collected in these databases. And I found that I just do a sort, and then found that there's an interesting distribution. So the top one is 1973. It occurred 2,500 times, about 70,000 different species. So all these different species share the same microRNA. So I think that's interesting. But the list, there are lots of other microRNAs appear in different species. So I think this will be an interesting topic for people who are in the field, that they can they know what the microRNA is, in which species, and maybe they can do experiments on these microRNAs in the different species, and they can add some of this microRNA to see if the mitochondrion behave differently. But this is outside of my expertise. But this is a summary of the most frequently occurred microRNAs in various species. So I think there's lots of work can be done from that. So the next thing I was trying to do is to search all the genomes and try to find the distribution of microRNAs in all the genomes. So human genome is 3.2 gig base pairs, and mouse genome has 2.7 gig base pairs. So I had the privilege to traverse both, looking for microRNAs in them. Okay, so let me see. A typical genome file of human chromosome 1 is 233 megabases. So generally in the data file, each base is represented by 1 byte. So the data file is 233 megabytes. So I'm just building my confidence in working with this file. So I have good tools to extract information from these big files. And I'm only using microRNA as an example. That I just do a search on the complete chromosomes. And in chromosome 1, I have found 7,670 microRNAs in the human chromosome 1. This is not many, because you can see the whole genome is 233 megabytes. And I can only identify 70 of them. And so I do the same sorting. And just one of the things that you want to know is how many of the microRNA exist and how frequent how frequent is their appearance. So here I have sorted and then arrange them with their frequencies. So I found that these two microRNAs occurred very frequently throughout the chromosome 1. And if you want to analyze a computer program, especially a high-level computer program, you look for call and return, okay? And I don't know. Maybe these are the call and return instructions in the cell computer. Yes?
A quick question. Okay,
yes.
If you have a call and return, that implies a...
I don't know. Okay. I just
like
assume.
a class and a stack.
Could be.
Could be. I find that interesting.
Yeah. I haven't found the stack yet. Okay.
Well, you know what? It could be transitory.
I don't know.
Yeah, that's what I'm saying is, and I'm assuming this is when the chromosome gets scanned, that there might be a, like I said, for a better word, a genetic stack. You can put, positions in it, so that you can go off, do something, and then come back. Yeah,
that's
it's transitory. You understand? It's
right.
You were saying in a previous talk that the code is juxtaposed with each other?
That's what I'm looking for.
Yeah.
I want to looking for instances where these microRNA occurred consecutively. And that looks more like a program. Right. But here in chromosome 1.
Right.
And then you know where it is, and it's not really a stack; it's just a return pointer.
Yeah. Okay.
But you can't.
All right. Well, let's change.
Okay. Yeah. Okay. So in the prior talk, I was talking about this. MicroRNA could be occurred in clusters, and that's what I was looking at. But in the chromosome 1, they spread out. So I found very few clusters, okay? But and they don't occur that frequent. So they don't seem to form clusters. Yes.
I want to bring up one point.
Yes.
Instead of call and return, how about spawn and wait?
Okay. All right.
So that you get
I don't know. But never mind this one. Okay, never mind this one. Okay. No. But it's just some of us doing reverse engineering. We look at the other people's code and try to figure out how he wrote the program. And basically, if you look at high-level code, call and return is the some of the key point that you want to identify. And if you find a call and find a return right next to it, okay, this is subroutine. You don't know what it's doing, but assign it a name first, and then find out if that routine was called from other places. So that's basically what I'm doing. Okay, so here is a list of the microRNA in human chromosome 1, and arranged according to their frequencies. So you can see this 26622, this microRNA occurred 4,000 times. And then the next one, 17325, occurred 2,600 times. And then all the others occurred much less frequently, but some of them occurred 40 times, okay? All right. So I just try to find some of the properties of the microRNA in various chromosomes. So I basically analyze all the chromosomes from chromosome 1 to chromosome 20, I think 22, and then chromosome X, chromosome Y. I have looked them all. And they don't look very interesting, okay? And I didn't find the clusters or the subroutines, but I just find them scattered all around. And so that's a human, okay? So I do the same for mouse, do the same for the nematode, do all the animals. And then I say, okay, I just go down the list. Since I have all the bacterial files, I just search the bacterial file. And lo and behold, couldn't find any microRNAs in the bacterial genomes. So I have the four different species that I look at, but I couldn't find any microRNA yet.
Didn't you say that Rudi, Bacteria, and E. coli are
single cell?
They're single cell, but no, it's the first two that are parasites.
Yes.
Okay. But E. coli can stand on its own.
Yeah, E. coli is then
that was the thing. Now,
freestanding.
I can see if the Rudi and the first two, then they would have to get a hold of some other,
the host,
a host genetic information in order to do this stuff. But E. coli can hang by itself. So, dang.
So I was very disappointed, and I almost gave up. You know, the foundation of my theory is that from the we call the messenger RNA. These are what I call the primitive code, and this primitive code was used to produce proteins. Okay, so we understand quite a bit of this messenger RNAs. So we have identified, in all this genome library, they try to identify every messenger RNA and then list them up with the proteins they produced. So, but if you look at the data, these are most of the things you see. But in bacteria, no microRNA. If it's true, my hypothesis of cell computers based on microRNAs as tokens will fail. Okay, so I was very disappointed. But then I thought, last week I was dreaming, and I thought, well, maybe the database of microRNA is not big enough. Maybe people have not studied the bacteria enough to identify the microRNAs in them and to have them listed in the database. So I thought that going back to the very early when I was first trying to look at the mitochondria and look at it and say, okay, 16,000 bases, but they are only most of them are coding DNA. So most of them are coded the protein and also the T transfer RNA and all these other things. So most of the function, most of the DNA in mitochondria, their function has been identified. And there's only one small section about 1,000 bytes, which are called D loop. And D loop is a stretch of DNA, doesn't code any protein. Okay, and people think, well, this is called a D loop or control loop, and this is where the function of mitochondria is controlled. But how this 1,000 byte mean, nobody really knows. So I thought that when you have the rudi and the Nashua, you got 100,000 bases, and then lots of the areas are non-coding, and so these things need to have some information in it, and what kind of information do you look for? And in my early study, I said, well, I just analyze, 4 base patterns, 5 base patterns, 6 base patterns. And when you go to 6 base pattern, the data table gets pretty big. 4 bases, each base is equivalent to 2 bytes. So when you have analyzed 4 bases, you have 256 entries. And you got one more, you got 4 times, and the table goes pretty big very fast. So I really don't at the time, I don't have the capabilities of looking at patterns, a much longer pattern. But after studying the microRNAs, I'm pretty sure that the patterns I have to be looking at about 20 bases or 22 bases, okay? And just to make it simple, I said, okay, I just look for 20 base patterns in this try to identify all the repeating patterns to see if they occur. And I think the recurring pattern function like microRNAs. Okay, so I wrote the program instead of having a fixed database of known microRNAs. I just say, okay, I got this big DNA sequence. I just look repeated patterns in this whole thing, and no matter where they occur. So what I did was just take the 20 bytes from the very beginning, and then move down to see if there's a match. If there's no match, I move down 1 base down, and take another 20 base pattern, and then to search. And if I search, I just record that base, and because well, you can go down further and look at all the matching pattern, but I think what I want to do is identify the recurring. So no matter if they just repeat once, I'm happy. Okay. So yeah, just go down and search the smallest genome. I found 150 recurring 20 base. Wait.
If you reduce the 20 base pair to the window to shorter, you can find more.
Yes. Yeah, if you reduce it smaller, you can find more. If you make it bigger, you will find less. Okay.
So the base, 20 is basically the microRNAs.
20, I think I could use 22.
I see.
But I use 20 just so that I don't miss anything. So 20, 20 bases. These are equivalent to 40 bits. 40 bits are a trillion.
Oh, yes.
Right? A trillion. You have a trillion different
choices.
Yeah, 1 trillion. So, the 20 bases search for 20 bases patterns is a good one. Okay. So the accidental match generally can be eliminated.
Right.
Okay. because there's only 1 in 1 trillion probability of mistake. Okay, you know, we can use 20, we can use 22, or the mix of it. But you know, this is just a study to see how many you can actually find. And for sure, you know, Nashua has about 100 kilobytes of base. So you have 156 recurring pattern. And the Ruda has 160,000. So the genome is bigger, so you can possibility of finding more matches are greater. But what do they mean? I don't know. I don't know. I think that's what I'm looking at. That's what I'm looking for, and I hope, you know, if these patterns form contiguous clusters, I'll be more happy of it. But I still don't have time to look at them, to analyze each and every pattern closely. Okay, so that's mostly what I did in the last 2 months. Okay. But I think you in the smallest single cell organism, there are very long, or not very long. These are the 20 base pair patterns.
I noticed that you have Python in there.
Yes.
I myself, with work and other stuff that I do, is I use Python. If you go to the Python website, www.python.org, they have up on the top bar something called PYPI, which stands for Python Package Index. If I'm not mistaken, there are genomic Python libraries that you can use, just so you have that in your mind.
Yeah. Okay. Yeah. Thank you.
You're welcome.
Yeah. So you know, these are the tools I've been using, and I rely primarily on F#. That's an eForce program that I've been used for maybe 20 years, and it serves me quite well in the Windows 7 environment. So I can open up the files and look at them, and then to manipulate the data inside. But I hit the ceiling in analyzing the you know, in the genome, in the human genome file. Besides all the chromosomes, there are also mitochondria, which is a small one, and they have a huge RNA file, and in which they collect all the known RNAs detected in human being. And that file is 500 megabytes long. And F# could not handle it. It handled all the files below 300 megabytes.
That's where Linux can be a little bit more helpful, because it can handle gargantuan size files.
Yeah, right. And of course, I didn't. F# was written by Mr. Chen in Taiwan, and he had already retired. He went to Thailand to be a Buddhist in a Buddhist temple. So he had retired, and nobody can find him anymore. So I have trouble in, you know, whatever he left in the F# package, I use, and the tools that he gave us handles the files up to 300 megabytes, and anything bigger, it failed to open the file. So in desperate, because the two RNA files in the mouse genome and human genome. In mouse genome, it's I think 350 megabytes, and I couldn't open it. And in the human genome, it is 550 megabytes, and I couldn't even look at it. And so I decided, well, I'll get onto Python and see if it can handle it. And in Python, I just wrote a program which would split a big file into 200 megabyte pieces, and then I can work with F#. But then the Microsoft Word, Microsoft Excel, and Microsoft Notepad, these are useful tools when you want to do, character replacement. Okay, Word's doing it really nice. Okay.
There are equivalents to those in Linux.
Yeah, sure. I'm sure. But I'm not a Linux. I have to survive with Microsoft Word. Okay. So these are some of the things. Excel is very nice if you can have a data file, you format it a little bit with Word, and then you can open it up in Excel, and you do the search real well. Okay. Sort according to one column, and you have many columns, and you sort it, and I just play with it, and I'm very happy with it. Yeah.
I was also thinking because when a DNA strand gets scanned, it's like a Turing machine.
Well, I think all computers are Turing.
Right.
Without the halting problem.
Right, but I was thinking, oh wow. So literally, it's like this is like a biological equivalent of a.
Yes, that's what I'm saying. Okay, that's what I'm telling you.
Excuse me, guys.
Okay. Okay. I think this slide will give you all the. Okay, the tools. F# works all the files less than 300 megabytes. And then I have some bigger files. I can split them. And you see the human genome is 3.2 gigabytes. If you want to work in this field, you have to be able to handle this 3.2 gigabyte file. Okay, but so far I see all the genomes, all the chromosomes are less than 300 megabytes. So pretty happy with that. If anything bigger, I use Python to split them up.
Really, you had a problem where Python was really slow.
Python is slow, okay. so you try to do things that Forth cannot do, and then once you can split things into smaller chunks, in Forth.
Okay, that's interesting. Because there's a computer algebra system known as SAGE, and where you can do a lot of math with it. And I've never heard of anything saying, "Oh, but Python is just so slow."
Yes, it is. Wow. But the genome basically is a text file.
Right.
So you just have to be able to handle text. And Python is beautiful in handling text.
Yes.
Yeah, you can big text, small text. You can search. You can look, and you can do all kinds of things.
Yes.
So the library, the Python library, is very very nice, very nice. But
you could add a memory map to I/O to.
Yes, that's a good idea.
Yeah. The problem with F# is that it opens a file in one of the two. It opens file and then open as a map, so it can read the entire file and drop them into the memory, and then you can change anything. If anything you change, it got stored back onto the disk. So it's very very nice. But okay, so I think I have one more slide that's one. Okay. So that this is a belief system. Okay. Okay. Yeah. So I believe each cell is a computer. And I believe the genome contains the complete code of the cell computer. So the genome has all the information for a cell to exist and develop. And I believe messenger RNA are primitive code of this computer, which produces protein. Okay. And I believe microRNAs are high-level code. And this code would activate microRNA and other messenger RNAs. So this is recursive, okay, and that's why I think it is fourth-like. Okay.
Cool.
Thank you.
There's a lot of stuff going on campus today, so it is recommended that we proceed with alacrity for lunch. It is.
For the record, I think this is a lot more interesting than what I was going to talk about today.
Oh, yes.
No.
Yeah, I have communication problems with Jay. What happens is sometimes Chromecast delays putting an email in my box.
Oh, yes.
So it scrolls off into yesterday's mail. It has the right date, but I don't see it because I don't look at yesterday's mail.
Exactly. I know that.
Okay.
Sometimes, mind you, I actually just delete it silently. So I never see it. There's no junk mail folder that I can look at. I guess they have one online if you use their website, which is perfectly bad.
A quick announcement before lunch. I brought in 3 tubs. I want to give it away. If you just want to leave money, it'll go for allocation stuff.
在这个里面，你这样搜能够应该比20 base pair 还长一些吧？
等一下，你再说，你再说一下。
就是如果在 Human Genome 里面你这样。
Sorry.
Yep. That's okay. Just want to make sure it stays.